Have you ever wondered if these plants are dyed, can the waste materials from them be used as a substrates in a fermentation? I think I need to ask these questions to Mr. Shakir if he have a solutions or any research finding about this kind of thing.
Hi, did you know that there is a high amount of waste worldwide from food processing industry which cause serious disposal problem? So, how the world need to solve this environmental pollution that caused by the food industrial waste? Therefore, I had discovered in scientific article, researcher or scientists found that one method which is solid-state fermentation that can solve this problem. This article titled Production of Ligninolytic Enzyme by Solid-State Fermentation Using Pleurotus ostreatus. This Pleurotus ostreatus is one type of white rot fungus. So, before I start, what is solid-state fermentation? Solid-state fermentation is growth of microorganisms on solid material in absence or near absence of free water to be utilized the potential of lignocellulosic waste. This solid-state fermentation was discovered the possibility of using byproduct and waste from food and agricultural industry. This microorganism can digest easily the waste because it rich in sugar which due to their organic nature. Therefore, these wastes are very suitable as raw material for microorganisms to produce significant secondary metabolite of industrial that useful in country. Therefore, in this study, potato peel waste was selected as most abundant waste of agri-industrial sector and world most wide-grown tuber crop and with largest food crop in terms of fresh produce after rice, wheat and maize. Besides that, prospective substrate for bioconversion into ligninolytic enzyme can be provided by using agricultural waste in bioprocess because of suitable chemical composition. Therefore, it can help to solve the environmental problem which caused by their disposal that can make the pollution. To evaluate peel potato waste as potential substrate for ligninolytic enzyme production, which are first, lactase enzyme, manganese peroxidase enzyme, lignin peroxidase enzyme and aryl alcohol oxidase by Pleurotus ostreatus. This method of solid-state fermentation can cause high production of lignocellulosic and other industrial important enzymes compared to conventional submerged. Solid substrate and its chemical composition, solid-state fermentation culture condition, analytical determination. This stage, we will use the potato peel waste as a substrate. We will take them and washing, cut them into pieces and will use as dry or fresh substrate. However, we need to implement the pre-treatment which is performed with potassium hydroxide or distilled water to neutralize the organic acid. We need to ensure that the culture condition is suitable for the microorganism for the fermentation to carry out. So, first is basal medium of culture. So, the basal medium of culture must be composed of glucose, its extract and copper sulfate. The medium also need to be adjusted to the suitable pH. And then, the solid-state fermentation process must contain 25 grams of potato peel waste as a carbon source. So, we will carry out this process in the 100 ml Erlenmeyer flask. We will take the actively growing fungus on PDA used as inoculant. After that, we will incubate the Erlenmeyer flask. So the sample from flask will be harvested, and the solid will be separated by centrifugation, and the supernatant will be used for the measurement of extracellular ligninolytic enzyme and hydrolytic enzyme. And then we will analyze the protein, nitrogen, and reducing sugar level. Stage, the important thing is we will analyze the nitrogen content using phenol hypochlorite method. The protein level also quantified using Bradford method. The lac activity enzyme will be assayed using ABTS method. So we will use this ABTS as a substrate and measuring its increase in absorbance. After that, the activity of manganese peroxidase also will be measured by using 2,6-dimethyltoxyphenol. The lignin peroxidase also measured by method of tin and Kerr following hydrogen peroxide dependent oxidation. Now, you must be wondering about the finding and result of this research, right? So, before I go even further, I would like to restate the objective of this study, which is to evaluate the potato peel waste as potential substrate for ligninolytic enzyme production by P. ostreatus under solid state fermentation process. So, based on the result of this research, we found that cellulose, lignin and protein content of dry potato peel waste were higher than those of fresh potato peel waste. In solid state fermentation process, higher protein level were generally found in dry potato peel waste used as substrate. We also indicate that usage of dry potato peel waste, which is treated with potassium hydroxide and distilled water, give the higher protein value, which is about 82 to 89.2 ppm on 7 days of cultivation, while the protein value of fresh potato peel waste only about 36 to 37.4 ppm. Other than that, the result also shown high production of other important enzymes, which are lactase, manganese peroxidase, lignin peroxidase on 17 days of cultivation, and aryl alcohol enzyme on 12 days of cultivation. This research has shown that the solid state fermentation technique have great importance as an efficient biotechnological process for Pleurotus species cultivation and the production of wide variety of enzymes. Other than that, we can also see that P. ostreatus has a capability to produce the hydrolytic and ligninolytic enzyme in fermentation of lignocellulosic material. From the fermentation process, we can determine that the usage of dry potato peel waste as substrate is the best because it contains high chemical content such as cellulose, lignin, and protein. In addition to that, peeled potato waste has high starch content and serve as an efficient matrix for the attachment of Pleurotus ostreatus, and this condition might due to high hydrophobicity and surface charge of the substrate. Besides that, the highest production of lac and manganese peroxidase enzyme activity in dry potato peel waste pre-treated with distilled water is associated with initial reducing sugar level in solid state fermentation process. From this experiment, we can say that the use of potato peel waste can be very promising for lac production as it might contain substances such as lignin, cellulose and hemicellulose acting as nutrient for fungal growth and inducer for lac enzyme. It is also proof that the content of lignin in potato peel waste increase when it is dry. Other than that, potato peel waste have a massive potential to make solid state fermentation process more economical due to their waste status, and it can also provide nutrient for the growth of microorganisms. the usage of potato peel waste as a substrate is very easy because it can process without adding any initial amount of glucose in culture medium. The researchers state that the usual manganese peroxidase enzyme synthesis by solid-state fermentation is hard to be detected when using other substrates such as corn bran, soybean, and wheat bran. We hope that the study of this potato peel waste will be broadened and applied to the industry because of the promising outcome, and it is so eco-friendly. As conclusion, from this research, it can develop an efficient strategy to minimize enzyme production costs, since this substrate seems to be more economical as solution for feasible alternative substrate. In addition to its affordability, using peeled potato waste as substrate in this study also contributes to the protection of the environment. It is very advantageous to obtain industrial production as well as saving energy and reducing waste production in financial terms. Therefore, utilization of waste on the one hand can provide high enzymatic level, and on the other hand, it ensures a considerable reduction in production costs.